Have you ever noticed that a tailor measures the size and then cut the piece of cloth with scissors? Keeping this concept in mind, let's discuss our topic of restriction enzymes. This is a type of enzyme, but it has a specialty in restriction. Restriction enzyme acts as an endonuclease. Endonucleases means this enzyme is used to cut DNA sequences. Consider this is a DNA. Its sequence will be cut by this endonuclease. It means this endonuclease cuts phosphodiester bonds between these sequences. A's in endonucleases is indicated for enzyme, as this enzyme is breaking nucleotide backbone sequence. So it is called nucleases. So, now from this we can conclude that restriction enzyme is simply the type of endonucleases. This nucleotide sequence is recognized by endonuclease, called as restriction enzyme. Means this restriction enzyme recognized this sequence of nucleotide and caused cleavage. For example, a particular restriction enzyme, or endonucleases recognize this sequence of nucleotide, GAATTC, sequence at a particular point, and causes cleavage, that is between A and T. So, wait, I have a question for you, and that is, can we use this enzyme for the welfare of mankind? And the answer is yes, of course. It is used in DNA, recombinant technology. We will discuss it later. So, from this discussion, we came to know two functions of restriction enzymes. One is recognition, and other is cleavage. Moving forward, bacteria have chromosome, called as bacterial chromosome, or bacterial genetic material. If a foreign agent, such as virus, called as phage, enters and attacks bacteria, it delivers its genetic material in bacterial cell. Bacterial cell degrades its enemy by producing restriction enzymes and degrade DNA of phage. So DNA sequence of virus is recognized by the restriction enzymes of bacteria and can be cleaved by the restriction enzyme. This is also the job of restriction enzyme. Because of action of restriction enzyme on DNA of virus, causes shortening of genomic material of the virus. But wait, there is a twist. This restriction enzyme can also cleave the sequence of DNA of bacteria, as the genetic sequence of bacteria is much more vast than the virus. So, the sequence cleave by the restriction enzyme on viral genome can resemble the sequence of the DNA of bacteria. It may be possible that this restriction enzyme may bind the sequence of the DNA of bacteria, and it can cleave its own sequence, but it cannot be tolerated by the bacteria, and it can be considered as a suicidal approach. So, there is need for modification of restriction enzyme, or called as restriction modification. So, it means in order to protect its own DNA sequence, bacteria needs a modification. And for this it has modification stamps. By using some units, it can modify its own DNA to protect its DNA from cleavage. Most of the time, This modification is through methylation. So, bacteria give signals that, "Don't cleave me. I have created you. Please cleave the DNA of our enemy." So, first function of restriction enzyme is recognition, and second function of restriction enzyme is cleaving the DNA of foreign agent, and third is modification of its own DNA to protect it from cleaving itself due to resemblance of its DNA sequence with sequence of DNA of its enemy. Moving forward, there are many possibilities. A restriction enzyme with recognition and modification, or restriction enzyme with recognition and cleaving property, or restriction enzyme with recognition, cleaving, and modification properties, or an enzyme with recognition and modification forms a complex with another restriction enzyme with cleaving property. Both after forming cluster works together. So this was all about restriction enzymes. In our next video, we will talk about nomenclature and types of restriction enzymes.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.